Right, everyone. Welcome to this Pan-Canadian Phage Therapy Forum, the first of potentially many. I am Alexander Hines. I am wearing my very best new dad chic as we have welcomed child number 6 to the household just a few days ago. So if I seem distracted, I probably am, but it's a real pleasure to help host this event, although really all the work behind the scenes has been by other people, and I get to take a little bit of the spotlight on their behalf. Now, before I jump into any of this, we are keeping this quite open format-wise. You have the ability to unmute yourself. You have the ability to do this. This is a risk we're taking. We would really like to have as much engagement and interaction in the audience as possible, but I do want to go over these session guidelines here at the very beginning. As you can see on the slide, we're asking you to use the little raise hand icon if you're familiar with this. If you mouse over somewhere on the Zoom, you should be able to raise your hand in the icon section as well. If you do speak up and you're granted privileges to speak, so we call upon you, do introduce yourself so we have some idea of where you're coming from and what your background is. But at all other times, we're asking you to be on mute. Now, it is worth noting that this session will be recorded, and that includes your question that you are asking. If you are not comfortable with this, of course, keep your camera off. Do not speak up. You just get to enjoy the session. But we do want to ensure that the people who couldn't attend today could also, benefit from the session as well. There are multiple ways to ask questions and interact, including the Zoom chat itself, which we'll be keeping an eye on, and the TriCider link, which you will have seen in the registration form and that we've been processing questions from. And I think there might already be a poll in the Zoom chat, but I don't know for sure yet, Daniela, whether that's been put up there. Looks like it has not. So there will be a pre-forum poll to sort of gauge where your knowledge is already before you come into this. And hopefully we get to see how that changes from the interactions with our panelists today. And I see Daniela scrolling around and so on. If we can't get the poll up right now, let's move on to the next slides and have some people speak. So very briefly here, we're going to have a quick few slides with some people introducing themselves and their interaction with Phage Canada or with the Phage community at large, so you have an idea of some of the people around here. So Daniela, could we get the next slide? Excellent. Oh, well, this one's an easy one for me. So I am presenting this on behalf of Phage Canada, which is a nonprofit organization that was sort of formally founded and had our first AGM last year and have a next one just in a few weeks. You can visit phagecanada.ca, and the mission of this nonprofit is to support and advance phage research and phage therapy in Canada across medical, environmental, and agricultural domains. I often say that this all started because of a Canadian Broadcasting Corporation CBC article in which it was claimed that there were only 6 people working on phages in Canada, which upset me for many reasons. One, that wasn't true, and second, if you count 6 researchers in Canada, you wouldn't include me. I wouldn't be in the top 6. And so, as an effort to coordinate the phage community and the phage interested community, I founded and formally co-founded with Dr. Greg German, who's here as well, Phage Canada as an organization. Right now, we have almost 120 registered and paying members, certainly more than 6. We're hoping to have a lot more by the end of the year as well. So there's clearly much more than 6 people and a lot of interested parties in bringing this research and this technology forward in Canada. And just as a shameless little plug, we do have a series of symposia on December 16th to 18th. These are largely showcases of research and happening in academic, industry, and government settings across the country with regards to bacteriophages. And so that's free for members. So I highly encourage you to sign up, and there's a registration link there as well. Moving on to the next slide. This slide here is a wonderful introduction to Dr. Steffanie Strathdee, who is joining us as a panelist and is working also from Toronto right now. She has an immense list of credentials, including being one of the most influential people in healthcare and that award. But I really want to share a much more personal approach to this because in the story behind The Perfect Predator, her description of a harrowing experience of her husband combating an antibiotic resistant infection and the phage therapy that she was able to seek out and help administer. This has been a huge game changer for the field, not necessarily from a technical standpoint, but I cannot count the number of people who have approached me because this is how they heard about phage therapy, because this is how it came to them. This, very visceral, personalized, and painful story, with a happy outcome, certainly, yeah, has marked so many lives, and I think in that way, it has had one of the greatest impacts on the advancement of phage therapy across the world. Just how many people it has reached and how many people it has been able to bring this forward. So it's an absolute delight to have her joining us on the panel today to speak about that experience and also as her role as co-director of one of the first phage therapy centers in the US, in fact, in the West. An immense amount of experience in the logistics and how to bring this stuff forward once it's conceptually possible. Next slide, please.
Sorry, let's let Steffanie maybe share a little bit more.
Oh, yeah, certainly.
Great. Thanks very much. I want to thank Dr. German and Dr. Hines for the opportunity to speak to you briefly today. I'm speaking to you from Toronto, my hometown, but for the last 20 years, I've worked at the University of California, San Diego, where I now co-lead the Center for Innovative Phage Applications and Therapeutics. But my foray into phage therapy began with the personal account that Dr. Hines mentioned, that my husband, Tom Patterson, acquired a superbug infection while we were on vacation in Egypt exactly 10 years ago. And he had been previously healthy, was exploring pyramids, and then the next minute he was fighting for his life. So you'll see on the slide here that there's a picture of him where he's not looking very well. He was in multi-system organ failure. This was taken in February of 2016 after he'd been in the hospital for about 4.5 months. And he's on a ventilator, 3 different medications to keep his heart working, and his kidneys were holding on by a thread. I came up with the idea of treating him with phage therapy after I looked into the literature. This was not something that I knew much about. I learned about phages at the University of Toronto, where I was a student decades earlier. And I approached my colleagues at the University of California, San Diego, asking if they could help me and if they agreed to maybe try this outside of the box kind of treatment that was a 100-year-old forgotten cure, at least in the West. And they agreed, but they said you have to find phages that are going to be a match for his bacterial isolate. Well, we were able to do that within 3 weeks with the help of a global village of researchers and clinicians around the world. And 3 days after he received intravenous phage therapy, you can see that he opened his eyes, lifted his head off the pillow, and kissed his daughter's hand. So our story was chronicled in our memoir, The Perfect Predator, a scientist raced to save her husband from a deadly superbug. You can go to theperfectpredator.com to learn more about the story. But to me, the reason why we wrote this book was because we were blindsided by antimicrobial resistance, didn't know anything really about how serious a problem it was until it hit our own lives. And phage therapy had been forgotten in the West. And so we wanted to bring that back. We've had enough success that our chancellor at the University of California, San Diego gave us seed funding to start the Center for Innovative Phage Applications and Therapeutics, known as IPATH. And we're a nonprofit associated with UC San Diego. And we've treated dozens of patients at our institution, but consulted on hundreds of others around the world and had over a 75% success rate. So I'm just speaking to you as someone who is a caregiver of a patient that received phage therapy, but now I'm an advocate for phage therapy because I see the potential and promise for this. And in my role at the University of Toronto, where I'm a visiting scholar at Unity Health Toronto, I work alongside Greg to try to show a spotlight on phage therapy to help and expand its access in Canada. And I'll be available for questions after the other speakers have had a chance to speak. Thanks so much. Greg, over to you.
Great, thank you very much. I'm going to save my comments for a little bit later. And I have a couple extra slides to share. I think we should go through our other list of speakers and then circle back to me. Dr. Michael Parsi.
Good morning, everyone. So, as Greg mentioned, my name is Dr. Michael Parsi. I'm the head of Phage Science, Therapeutics, and Research with the Public Health Agency of Canada. So we are a federally operated phage susceptibility testing reference service that was funded quite recently in 2023. So the goal is that we have this centralized phage collection that Canadian clinicians are able to access and send us clinical isolates that we match against what we have here. Currently, our collection is about 160 E. coli phages that we've isolated using our carbapenemase producing E. coli panels, as well as 43 Pseudomonas phages that were donated by Dr. Laurie Burrows at McMaster. One of the great positions we have working out of the NML, which is Canada's high level containment facility, is that we have national wastewater surveillance programs, national AMR and nosocomial infection surveillance programs, and we're able to tap into that material to do high throughput and rapid isolation for phages that are specific to clinical isolates. And so with that, we are using new panels now to expand into Staph aureus and Club pneumo as well. So we've soft launched our phage susceptibility reference service. That is to say, we've reached out to clinicians and they've been able to send us clinical isolates, and we're going to be testing our collection against what they're sending in, as well as isolating new phages to kind of fill in the gaps. And the goal of this is that we'll be able to launch this program next year. So this will be through the Guides to Services with the Public Health Agency of Canada. It's a collection of reference services that are free to Canadian clinicians to access. And from that, we do the matching, and we're hoping to be able to send the phages back. Currently, the phages that we'll be responding with will be as is, meaning they won't be in a purified state, and we'll have to work with the clinicians to move them into that therapeutic realm. But we're really excited to be able to do this and put together this centralized collection. And I'll be sticking around to answer questions and anything in the chat. Thanks.
Thanks so much. And you set the pace for everyone else in terms of timing and coordination. It's great to bring that national Canadian spirit. And it's also not just you, but a big group of interest of seeing how we can have the National Microbiology Lab be not just Canada's phage lab, but an example for how it should be done at the international stage as well. We can move on and we'll circle back for questions. Dr. Richard Siggers from Health Canada.
Hi, everybody. Thank you very much for the opportunity to join this meeting. I'm going to do just a quick overview of Health Canada's experience with phage therapeutics. To date, we've had approximately 10 clinical trial applications, and these have only been for the open label single patient of variety. We are anticipating to get some more kind of traditional clinical trials, but so far we've approved approximately 10 clinical trials. I do want to touch a little bit on Health Canada modernization events that we're undertaking right now. And I'd like to draw a little bit of focus to the modernization that has occurred so far, and it has been Division 2 and Division 8. And those modernizations and those updates to the regulations have come into effect already, come into force. And Division 4 was anticipated to be done by this December. I think it will be coming shortly thereafter, but a lot of the legwork has already been done for that, and the public consultation has been coming. So these are some modernizations, and a lot of these modernizations really allow for greater flexibility. We are currently undergoing the clinical trials modernization for the Division 5 regs. And I'm not entirely sure where that's at. I think it's just strictly internal right now. So a lot of the legwork is being done now. So you can anticipate that's coming out in a little while. And then also one thing that I would like to draw attention to is a key component of our modernization and increased flexibility is the advanced therapeutic pathway. And that came into effect in 2019, the ability to use this pathway. And we are currently internally looking at different pilot projects to run through this ATP pathway. So just a little bit more on that. What is it? It's really designed to give flexibility to Health Canada and to sponsors who are bringing new and innovative drugs and medical devices. So basically anything that's not really fitting into the traditional framework of our other regulations, we can build and adapt a regulatory framework and conditions around these products. For example, AI or medical devices. And I actually think that there is a lot of potential for, depending on the path forward for bacteriophage, that this may be one of the tools in the toolbox that we can use. And it is important to note that although we have this flexibility, we still are going to we still will maintain the appropriate oversight and ensure the quality and effectiveness and safety of these products. So really just as a closing message, we've done a lot of work in Health Canada to really update and modernize the regulatory framework to allow for the greater flexibility. So really now we just need to know where is the need and why is it needed. So I'll finish with that closing statement. Thank you very much.
Great. Thank you so much, Rich, for everything that you're doing. It's a big team effort from Health Canada, and we're in the process of looking at our second case right now. Dr. Gina Suh from the Mayo Clinic will have two announcements, and she's my colleague here. And so I'm just hoping that we can make sure the video is appropriate. 1 second. Okay, we're good. And then just trying to make sure I have the most appropriate view. Ready for you, Gina.
Hi, everyone. I am not Canadian, but hopefully I can be an honorary one today. Two quick things. First, I wanted to make a plug for Asgenta, which is a study group. It's the ESCMID study group for non-traditional antibacterial therapy. And it has now become quite a large group with more than 850 members as of yesterday, representing 121 countries. And it was established by Dr. Ron Nearpaz in 2021. And it's a study group that covers all aspects of phage and lysine related therapy. And I think really it's more than just a study group. It is a way to give us, the phage community, a voice, and to get together and have a seat at the table and create the agenda that we need to bring our field forward. So we have a bunch of activities and conferences lined up for 2026, which I've listed there below. Please join, participate, run for executive committee. There's going to be a new set of elections in the spring. Next slide. And then just real quick to give you an idea of what we've been doing in Minnesota at the Mayo Clinic. So I run the clinical phage program at Mayo. We've treated about 20 or so patients with phage and 2 of them with lysine therapy, finally, which I'm super excited about. We've treated 2 patients with prosthetic knee infections.
Gina, what's a lysine?
Oh, sorry. So lysins are phage adjacent. They are the enzymatic end product of phages. So it's the actual chemical enzyme that lyse the cells. And so I kind of tell people it's like the phage is the snake and the lysine is the venom. And so we've gone right to the venom and used that directly in a couple of patients, and it's too early to talk about results yet, but that's been very exciting. But a handful of patients that we've treated, we've learned so much with every single patient, and it's just been exciting to contribute to the clinical applications, the writing protocols, developing protocols, figuring out how long, what dose, what route of administration to treat these patients. We don't have all those answers yet, but I think as a group, we're getting a little bit closer. So thanks very much, everyone.
Okay, great. We're doing a swap out now. If Danny can stop sharing her screen, Dr. Tristan Ferry from the University of Lyon will be giving his perspective, and he's such a champion. Last year, right around this time, my son, myself, and 5 others from a Canadian delegation were at a workshop that he put on, and it's with great pleasure to have him also speak for us today on this Pan-Canadian Phage Forum.
Thank you. Thank you so much, Craig. Good evening, everyone. So I would like just to introduce you the Phage in Lyon clinic program. That is one program in France to develop phage therapy. So I am an infectious disease physician, and my aim is to expand the access of pharmaceutical grade phage and lysins. So we treated close to 100 patients with phages, 19 patients with lysin, with different setup of complex bacterial infection in compassionate use program, but also in clinical trials. We are receiving a lot of phage requests. So here is the map of the location of the physician who are contacted us to treat the patient. And most of them are complex bone and joint infection because we have a dedicated surgeon and a referral center for the management of complex bone and joint infection. And the French Healthcare Authority gives us the ability to centralize the request at the national level for these patients. And we have a dedicated clinical flow with dedicated pharmacists, but also dedicated radiologists, orthopedic surgeon, infectious disease physician, microbiologist. And we are doing each week a multidisciplinary meeting to discuss potential indication of phage therapy, because all bacterial infections are not relevant for phage therapy. As you can see, we treated most of the patients we treated have bone and joint infection, and we have in France involvement of the French Healthcare Authority with a French law that facilitates now greatly the compassionate prescription framework and the compassionate access authorization. And we have a dedicated team to validate the indication. We are sending the strain to the phage producer. We are working with companies, but also military setting, and then we order the phages. If you're thinking that some of your patient or yourself have complex bone and joint infection, and if you would like to ask us, it's possible also to contact us outside complex bone and joint infection to give some advice. So don't hesitate to check our website and connect us on LinkedIn. Thank you.
Thank you so much, Dr. Ferry. And we'll have you unshare your screen so we can go back to the... Very good. Dr. Antonia Scooby from the United Kingdom would like to share more about what's happening there. And it was with great pleasure that I was able to go and see her recently at a Ministry of Defense / Research Council joint discussions on how we need to move forward quickly together.
Thank you, Greg. I'm Antonia Scobie. I'm an infectious diseases physician and microbiologist based in the UK in London. It's safe to say that we are a fair bit behind what's going on in some of the from the international groups that you've seen already. So the situation that we've been in is that we've had sporadic cases of patients treated with phage. Most of the time they've been imported from other countries. And there are a number of reasons why that we've been held up. And as you've seen with some of the Canadian groups, we're working very hard to address some of these gaps and obstacles that will make it easier for patients in the UK. So we have developed a group called Phage UK, and thanks to Greg making a bet with us that we couldn't come up with a logo before Christmas. Here is our logo. Thank you, Greg. So this is a group of clinicians and academics and scientists who are all dedicated to advancing phage therapy in the UK. And thanks to a lot of the groups on this call, we've been able to actually follow the example of what's been going on internationally. So we still have a number of sporadic cases being treated, and we think probably around 20 cases across the country so far, but we are working towards developing and being involved in a number of international clinical trials. But we've developed working groups amongst a lot of the areas that we think will benefit from phage therapy. And through these working groups, we've actually formed a network that we can review cases that we think would be suitable for phage and help other clinicians work out how to access phage and deliver it to their patients. The other thing we work towards is having a national registry so we can collect real-time data on patients that are referred, considered for phage, and then go on to receive phage. The issues we've had are lack of regulatory guidance and structure, and the fact that we had discrepancy between what you could import and whether a phage needed to be manufactured to a certain standard in the UK. And thankfully, there have been some recommendations that have forced our regulators to at least provide some clarity. Not that it makes it a huge amount easier, but we're working with the regulators to make that easier. The other thing that we are working towards is building on the infrastructure in the UK. So trying to access phage testing, which will be through our health, our public health system. So UKHSA are developing a phage service similar to what was presented in the Canadian spectra here, and also working towards a national funded service. Next slide. So this is just the group that we have created and the pathway that we've a number of clinicians who are interested and are trying to lead their efforts in their respective fields of areas that we think would benefit from phage therapy. So we're happy to be approached. This is the website of Phage UK, and also receive any patients that people think are considering for phage therapy in the UK. Thank you.
Now we have Dr. Daria Van Tine from the Pittsburgh Phage program that would like to share what's happening a little bit down south from Canada right now.
Hi, everyone. Thanks a lot for the opportunity to tell you a little bit about the Pittsburgh Phage program. On a personal note, my sister recently emigrated to Canada. She and my brother-in-law live in London. But we're just across Lake Erie from them down in Pittsburgh, Pennsylvania. I'm at the University of Pittsburgh in the Division of Infectious Diseases. When I came to Pitt, we started the Pittsburgh Phage program. And the engines of the program are my lab as a phage lab, and also Graham Hatfull, who many of you know, also a phage lab here. So we've sort of joined forces, and you can see the many people, other people besides us that are involved, and many of them do the actual work. Our target organisms are on the top right. So the left-hand column are the potpourri of organisms that my lab has phages for, and then all of those M organisms are mycobacteria that are targeted by Hatfull lab phages. The patients that we've screened for phage susceptibility include a whole variety of patients with underlying conditions. And then the bottom right are just some quick facts for the current volume and what we've done so far. So we're coming up on 1,000 patients that between my lab and Graham Hatfull's lab that we've screened for susceptibility. I'm sure we're well over 1,000 isolates screened, but many patients have multiple isolates. So the per patient numbers is not quite 1,000 yet. But we have treated over 100 patients between our lab and Graham's lab. And those patients have been here at the University of Pittsburgh Medical Center, but also all over the United States, as well as a couple of patients internationally as well. So I'm very happy to be here with all of you today, and looking forward to hearing the great discussion. Thanks a lot.
Thank you very much. We have Dr. Ibrahim Barbari that is wanting to offer a perspective from Saudi Arabia as well. And then we'll all then go to Phage Australia after that. I'm also happy to share Dr. Barbari will be a fellow in microbiology and chronic infections, focusing on urinary tract infections as well as orthopedic infections at the University of Toronto and the Unity Health Toronto hospital system. That's scheduled to start in March, but I'd like for him to start with his perspective on what's happening where he's currently located in Saudi Arabia. Please.
Thank you, Dr. Greg. Hi, everyone. Thank you for the chance to speak today. I'm an infectious disease specialist, currently based in Saudi Arabia with a personal interest in phage therapy. And I just wanted to give you an update of the state of phage therapy over here. We are unfortunately a bit behind, with no cases of therapeutic uses of phages in human patients as of yet, and no experts on the clinical side of things. What we do have is a community of scientists who have worked on research and biobanking. The most prominent example of their efforts has been in my own hospital's research center in National Guard, where we have a library of a few hundred phages targeting local isolates of Pseudomonas and Acinetobacter species. Phage therapy, fortunately, does align with our own Saudi National Biotechnology Strategy, which does mention as one of its key goals the localization of biomanufacturing, especially of more advanced drug products. So we are now attempting to leverage this as we apply to an NIH grant. If we do manage to get funding for this and things go smoothly with our FDA, we could have our first administration of phages to a patient around mid-2026. Now, like many areas in the world, we have the issue of navigating our legislative bodies. So the trial designs we had in mind were one, a phase 1, phase 2 trial with safety as the primary endpoint to help us basically get our foot in the door, or we can maybe get a bit of a shortcut using what we have that is referred to as the accelerated use program. That's basically our equivalent of the compassionate use program. Now, for the gap in clinical experts, I hope to get to tackle this with my fellowship starting in March of next year, where I'll be joining Dr. Greg. And as mentioned, we're going to be looking at patients with refractory infections, limited treatment options, and possibly offer phages if suitable. With that, I thank you for the opportunity. I look forward to hearing everyone's questions and input. Although I would like to mention, it is also an honor to get to speak in front of Dr. Steffanie. As many, I was initially inspired by her book, and her initial setting actually in Egypt is literally less than a 2-hour flight away of where I am. And thank you so much.
Thank you, Debarbari. Appreciate it, and look forward to seeing you soon in Toronto in March. I'd like to now have us not share the screen on our side and then have Dr. Ruby Lin from Phage Australia present. I was there again about a year ago, and with great hospitality, Phage Australia has been one of the forefront organizations and an excellent mixture of solid microbiology with solid infectious disease. Dr. Ruby Lin. And I also happen to share with you that it is a little bit early in her time right now, so she may elect not to be on video, and I'm okay with that as well.
Can you see my screen?
Not yet. It's in progress. There we go.
You see it? You see it now? Good morning, everyone. It's 3 o'clock in the morning here in Australia. So Phage Australia is a not-for-profit organization, and it's based on the STEM protocol, which is Standardized Treatment and Monitoring Protocol. We managed to get national ethics approval in 2021, and this is built on clinical trial oversight. It ensures the quality. We produce our own phage in a clinical research setting, and this we set it up as an integrated system to build trust amongst the prescribers, consumers, and regulators. And of course, we're able to systematically collect specimens, collect clinical outcome. And it's at the moment we've got 59 hospitals around Australia. That includes major teaching hospitals as well with a site-specific approval, and 19 sites are actively treating patients with phages. At the moment, the phage are produced under GMP exemption, and it's through TGA Special Access Scheme. and it's made here at Westmead Institute. We work very closely with our regulatory body, Therapeutic Goods Administration, as well as the OGTR, which is the genetically modified arm of the regulatory body. In October, for those who are in the know, TGA put out a public consultation paper on GMP exemption, and they just want to make sure where in the Phage process pipeline that we can actually place GMP. And there's a paper here, I just put out paper here that we published about the STEM protocol.
Thank you. And if we just need you to either share another slide or stop sharing so we can go back to our slide deck.
Can you hear me now?
Yeah, we can hear you.
Okay, I'm trying to go to the next slide. Okay. So far with the STAMP, we have under the STAMP protocol, we've treated more than 40 patients. So this is the data for 34. In total, we have treated over 60 patients by this team. We have produced over 67 batches, 26 phages, single use as a single phage as well as the cocktail. And you can see on the right-hand side, it's a combination of treatment as well. And the phage used in the treatment, so these are the different pathogens that we have counted. And this is our network.
Amazing. Thank you so much, Dr.
Thank you.
Okay. We'll go back to our slide deck. So I'll be presenting on behalf of Dr. Ben Temperton from the UK, who also has some Canadian roots. And what has been fantastic is this outreach at the citizen level of having samples sent across the UK and other countries to obtain samples from the environment. And you become a certified microbiologist by doing this, where you end up collecting samples from ponds, from runoff. Runoff is the nicest way of saying certain things, but from runoff, and then you actually then purify it of the bacteria by in using a filter, and it's that filtrate which has the phages in it. It's interesting that phages were first identified by being so small that they would go through a filter while bacteria would stop, and then you end up sending it off. And so they've done this a great deal already. You can see here over 800 citizen samples, over 6,000 wastewater samples, 1,300 phages isolated against the WHO, World Health Organization priority pathogens. They've supplied phages to Belgium, Australia, USA, Canada to support their phage therapy efforts, for which we're grateful. And they've been working with the UK hospitals and manufacturing governments to unlock barriers to use. So this is fully automated with AI-assisted assembly and annotation. So, getting the genetic sequence, the DNA code, and using ways of interpreting that quickly is what they're talking about there. And sampling campaign is coming to Canada in 2026 in partnership with Phage Canada. So Ben sends his best, and we look forward to hearing more about this next year. Next slide. I'd now like to introduce maybe future doctor someday, Daniela Parisi, in regards to the University of Toronto Phage Therapy Interest Group. And I'm also happy to share that that Lena Siddiqui is a master's student in our laboratory outfit that's also quite involved with this group. Daniela, please.
Yeah, so just a quick intermission from the amazing talks we've heard so far. I'd like to briefly highlight the work we're doing at the student level to grow interest and capacity in phage therapy. So hi everyone, my name is Daniela and I'm one of the founders of the University of Toronto Phage Therapy Interest Group. We are a student-led initiative aiming to inspire the next generation of scientists working in precision antimicrobial therapies. And our main mission is to spread awareness about phage therapy and how it moves from bench to bedside and highlight scientific, regulatory, and clinical challenges to make this feel that highlight this field that's both complex and exciting to work in. Before I continue, I want to quickly introduce my co-founders. Lena, if you want to go ahead.
Hi, everyone. My name is Lena, and I'm Dr. German's master's student, like he mentioned, at the University of Toronto. My research mainly focuses on phage susceptibility testing and figuring out how to do this in a clinically relevant time frame. And I mainly focus on urinary tract infections. My undergraduate background was with carbapenemase-producing organisms, so this field of phage therapy was kind of like a natural transition for me in terms of treatment options. I'd like to thank you all for attending our forum today, and I can pass it off to Taranveer.
Yeah, so my name is Taranveer Gotra, and I'm currently a fifth-year undergraduate student at the University of Toronto studying life sciences. And I became interested in phages and phage therapy after learning about the overall global rise in antibiotic resistance and the urge of finding alternative treatments. And that's when I came across Dr. German's research, which uses phages, which can target antibiotic-resistant bacteria, such as those responsible for urinary tract infections. And then his work has inspired me to dive deeper into how phage therapies are developed, tested, and translated into treatment as well.
And back to me, a little bit about me is I'm a third-year undergrad and a research assistant in Dr. German's lab. I've previously worked on characterization of 2 novel phages that have been GMP produced for upcoming compassion use trials in Frankfurt, Germany. And currently I'm helping with the Canadian phagistry development, which Dr. German will talk about later. So for our group, we have a few key initiatives currently in the work. So first, we would like to host coffee chats or seminars with experts who work directly with phages, because we think that's a great way for students to learn directly from people who's actively shaping the field. We'll also be running a journal club where we'll review landmark papers and discuss current gaps in phage therapy. And finally, we would also like to put some focus on outreach and collaboration, reaching out and partnering with various groups involved in phage therapy within Canada to spread awareness and help advocate for phage therapy. As we're just starting off, we're currently expanding our University of Toronto team and looking for students who are curious about phage science and antimicrobial research. We're also open to collaborations with other universities, student groups, or anyone interested in building a phage-related initiative. If you're interested, you can scan the QR code at the bottom of the slide to sign up, share ideas, or just leave your contact information for us to reach out. And lastly, feel free to follow us on Instagram at UFT Phage Therapy. We'll love to have you guys part of this growing community.
That's fantastic. I think there's maybe one more slide for me. How are we supposed to do this? And in Canada, when we only have 10 and it takes upwards of 18 months to have another, how are we supposed to quickly, rapidly focus on good data? And one process is developing a registry of clinical cases that will allow people to get quick and rapid access to phages and having their isolates, their bacterial isolates, sent to laboratories that we've already talked about today. And so there's an international initiative to do a phagistry, which is like a registry for phages. And that's been going on since 2021, and we've been an early participant in it, but it is with great interest in 2026. We need to move forward to have it so that this is a go-to mechanism for looking at phage cases while they're in progress or afterwards, so we can collect solid safety data. And it's with that solid safety data that we can work within our other partners, including Health Canada, to potentially have it so that it doesn't require the great effort that is, because we've found through other studies, phages are particularly safe and effective, or at least particularly safe. How do we prove that safety? How do we monitor that and make sure it's not just, everyone's best case, but the cases that don't work, we don't hear about. So it's with interest that to make this more of a Canadian node or initiative within Canada, located at St. Michael's Hospital and their academic research organization unit, to be able to participate in the Canadian fashion and then support international data that will help regulators and will help interests as a whole on seeing and proving that things are, and that phage is both safe and effective. So this is from Dr. Shauna McKellen. She's in Switzerland right now and has had excellent connections. She herself is involved with focusing on urinary tract infections, and they've even talked amongst our groups about utilizing stool transplants and phage therapy at the same time to try to rewrite the bug code in someone's body so it's no longer antibiotic resistant nightmare organisms, but actually back to baseline organisms. So that's what I wanted to share from a phagistry perspective. I'd like to, I don't think we have any other specific slides showing, is that right, Danny? Give me a thumbs up. Well, we've done pretty well so far. 45 minutes in, we are halfway done of this, and we haven't really gotten the chance to really engage with other people, but there has been engagement happening behind the scenes with questions that have been posted, and we'll be working through those questions in just a second. Through those questions, if something happens, we'll end up going into my slide deck as things come up, and it'll give you a little bit more of a history or background. I could spend enough time talking about what I would think would be important, but we really want to answer your questions or at least do our best to answer those questions. So first things first, we are going to do a pre-test, and it's a little bit unfair in part because you've already heard what's going on, so you may know more, but we at least want to get everyone's idea of how familiar you are with phage therapy. And maybe you want to take the notion of before what you saw the last 45 minutes. You can change your answers if necessary. We have over 66 people signed up and participating right now, which is fantastic. And I'm seeing people of all walks of life, all parts of the world, many, many, many from Canada. You may have even saw that word cloud. It's gigantic Canada in the middle. Great job. That means there are a lot of people there. But at the same time, there was at least 20 countries from around the world that have signed up for this Pan-Canadian Phage Forum, which is the first of its kind, at least that we're aware of. There has been excellent forums on African Phage Forum, which I've been able to talk about, but we have the general public here as well. We have people who are from the media, as well as people who have donation history. So there's a lot of potential opportunities and angles. So we're going to now at 46 people who have participated. Let's see how I can get this end the poll, share the results. You know what? We have a really super interesting group here. We have 60% that are very familiar, 29% that are somewhat familiar, and then 10% that heard about it. So we'll do this again near the end to see if we've changed anything. And Lena and Danny, is there a way that you can take a quick snapshot of remembering these things so we can see how much it's changed from before and after? That would be awesome. How's that? Are we able to give a thumbs up if you took a snapshot? Okay, great. So we're going to stop sharing now. Danny, I think it would be great if we start going into the question-based format. So maybe if you want to stop sharing this, and then I think it was the Google group organization that was probably one of the best ways of doing it, or it was the TriCiter. It's a little bit up to you on what you think what would be the best use. We may end up flipping between the two, if that's what you'd like to do. In the meantime, there's some of you that this is an open forum. You're allowed to raise your hands. Of course, we'll call on you, but you can see that there's a chat as well. And if you'd like to introduce yourself, and maybe today you were on the fence of presenting a slide or whatnot, but if you'd like to provide a little bit more of a feedback on where you're coming from and what your interest is in the Canadian space, or even from a phage outside of Canada, I encourage you to feel free to try to write a haiku or write a couple lines about what you're coming through from a today perspective. I'm going to start sharing my slides and talk about other things, Danny, or would you like to go with the questions?
We can start with like a question. One of the main questions, or a common question I saw, is like, what's the projected timeline of phage therapy within Canada? Any updates on that, and how you see it projected into the future as well?
It's almost as if I want everyone's point of view regarding that. It is a complicated question. There are people already getting phages from outside of Canada, and we actually had Canadians going to the US and Yale before they were actually treated officially through research studies. Further to that, there are people who are going to the Republic of Georgia, or somehow those phages are actually, because they are provided without needing a prescription in Georgia, but they follow manufacturing processes that are pharmacy or at least food related. They are being used and sent back from a phage perspective. So there are a lot of people in my clinical space that are aware of those products. So that's one way to go for it. But if we're really talking about how do we make it a prescription product, In Canada, it requires multiple steps. And Dr. Richard Siggers could probably add more to that. I would offer that as a physician, there are certain special access pathways that once there's an official licensed product that has what we call a drug information number, and that's done internationally. And as long as it's approved by another regulatory body, the United States or the Europeans, then we can essentially write for that to be brought into Canada. So at this time, we don't have that product yet. We're probably 2 years or more away from having one of those products available. There has been some clinical trial support, which will help. But when we think about research, you have to have phase 1 studies, phase 2, phase 3, get enough phase 3 data together to make a claim, and then those claims are endorsed by the regulatory bodies and allowed to market that medication, or in this case, the phage, and then provided with the use of a prescription. So I would say that we are maybe 2 to 3 years away from using the special access process pathway, and I think we're about 5 years away from having a made in Canada solution that can be marketed within Canada. I also think, and I'm going to not try to take up too much more of the time and allow other people to answer, there may be other methods, including use of compounded phages, which is like an alternative, more provincial way of having access to phages that we can talk about. And then finally, if we can do something that, let's just say, puts things in a sandbox, a regulatory sandbox, where we don't have to do all these special applications that it takes tremendous amount of time and energy, including research ethics, for a well-defined pathway. And I think that phagistry is part of that pathway. We could have access to phages with the use of infectious diseases within the next 18 to 24 months, which would actually show that Canada can come to the plate and hit way above its weight in utilizing this unique pathway. And we've actually done that before for fecal microbiota transplants, which we used to treat C. difficile. Now, phages and C. diff are different things, obviously, but we're learning from it. And it's episodes and sessions like this that show how important it is that we get our butts moving. I'd let Health Canada or anyone else that would like to add more to that question.
Thanks, Greg. Excuse me for not raising my hand. I couldn't actually, I can't figure out where it is in this, so I'll just unmute and go. Okay. For sake of time, thanks, Greg, for that overview. And maybe what I can answer is some specific questions if people have them after. But I would like to make some comments about the regulatory approach. I think it's important to note. I think Canada is very well positioned from a regulatory perspective to be very amenable and flexible. I think we have all the components. I think it's with great interest you see some of these the presentations and the approaches, for example, STAMP that Ruby just presented. These are all very real possibilities here in Canada. And I think really what I think the message that I really would like to deliver to the group here is that there needs to be an initiative and there needs to be a collection. I think the difference that I see between Canada and Australia right now and France, and the nice presentation done regarding the facility in Lyon, is there's a very big coordinated network. And so once that's established in Canada, then the collective network can come to the regulatory agencies or the people that are making the regulatory decisions and say, this is what we were going to propose. And then we're very well set up from a regulatory perspective to have these discussions and to work through the regulatory framework so that we can achieve what the objective is. I think what has been done in Australia is a very good starting place and an example. It's published, it's available, and so I think that's a good template for starting. As Greg did mention, we do have the flexibility even outside of our modernization. We did with the FMT. And I think that's a very good success story. So I think the first things first, I think there really has to be an effort within Canada to set up a similar type of network. And once that network is established, then come to Health Canada with a proposal. This is what we would like to do. These are the nuances of what we'd like to do. And then that starts the discussion. And then we have a starting place and we can really move forward. Now, so there are some differences, and Greg, you mentioned about Georgia and prescriptions and stuff like that. We have to remember too, once it depends on how we're classifying the drug and the phage. So phage, if not considered a drug or doesn't, isn't going after a particular indication, we're more in line with probiotics. And that's a different regulatory pathway. And I think that's what it is in Georgia because they're giving mostly topical or oral. So according to our framework, as soon as we puncture the skin, then it becomes a drug and then it becomes a little bit different. So there are nuanced differences as well as how we classify a drug. And it's based on it is specific for this indication. And once we attach a clinical indication to it, that becomes a little bit, there's a higher burden of regulatory proof there and more steps. So I just want to highlight those are specific differences. As far as the compounding, again, Greg, you just touched on it, but I think that was a point that's maybe hasn't been clarified or well understood. The compounding is different, and that's regulated at a provincial level and under the College of Pharmacy, not at the Health Canada national level. We have to approve the manufacturing of the particular phages, ensure that they're controlled. But if they go to a pharmacy and they're compounded, like in Belgium or in Georgia, that's under a completely different regulatory body. So we have to take different considerations about that, or the physicians and the stakeholders have to consider those differences. So those are some of the things I just want to, I could go on for a little bit longer, but I don't want to take up the time. But I think those are kind of some comments I'd like to put out as discussion points right now.
Thank you so much, Rich. I would take an opportunity to, I was part of the AMI Canada Phage Therapy Working Group as its chair for 3 years, and I have handed the reins over to Dr. Marek Smedja. And would he like to address the question? If not, maybe you could put some comments in the chat as well. Marek?
Sure. Can you hear me, Greg?
Yeah, we can. Go ahead.
So I'm just recovering my voice. So I've participated in the last couple of AMI Canada meetings around this. So my job in terms of chairing the working group is really to bring together Canadian, especially infectious disease physicians and microbiologists interested in phage therapy, interested in developing some of the infrastructure for how we would give this to patients in our clinical centers. We think it's primarily going to be infectious disease doctors, maybe orthopedic surgeons, maybe a few other groups who are going to be primarily involved in identifying people who are difficult to treat and for whom phage therapy may be helpful. We're still very early. I have not personally done phage therapy. We're still very early into assembling how we can make sure there are some Canadian and or international people to give advice on when it might be worth going down this way. And then I think, Canadian ID physicians are going to need a lot of help in terms of how to maneuver this. I think realistically, unless people have an independent research program that you can use people from, I don't think a busy practicing clinical ID doc today has any way of getting involved in phage therapy directly. So we're realistically going to have to identify where people have both clinical but also research infrastructure to help with this, because I think realistically it's way too complicated at this point for most Canadian physicians. But hopefully in the next few years we'll work towards how we can as a group identify the barriers and simplify them.
Rich?
Yeah, this is great discussion, and maybe I'd like to take the opportunity of having Ruby online and Ruby, in your experience, I mean these difficulties in setting up a network. This must have been one of the hurdles that you had to overcome. So maybe if you could take a few minutes and just kind of highlight. Were you experiencing these same difficulties and what were the biggest hurdles in setting up a network? And what was as far as the events, the chronicity of the events, did you set up the network first? Did you then engage with the regulators? What was your experience and maybe what advice would you offer the collective group here and how we could expedite any process to maybe work towards something that you've established in Australia under the STAMP protocol?
Well, thank you. When we first started, John Iredale actually treated a patient back in 2007. And so we've always been interested in phage therapy. And as far as talking to the regulators, we've been engaged with our TGA as well as the genetically modify OGTR for at least over eight years. And we were very lucky because for those who are not aware, FMT Australia was actually one of the first ones, and we've published a paper on FMT protocol. And so the TGA is very permissive. And from that perspective, we know that we've got supporters that would actually help us identify industry best practice, and therefore they can actually formalize the regulatory framework for phage therapy. And I made a few comments while other people, I think Merrick was talking about integration. You need funding. We spent a lot of the research funding. We got grants and all of that stuff, but all of it was going into personnels to look at the pipeline. So from in the slide that I showed, from the beginning of extracting phage from the environmental samples all the way down to matching, susceptibility testing, to phage production, and even from all of that stuff, you cannot have one person doing it. You actually have to have an integrated team to do that. So we actually have carried out 4 different health economics analysis to look at, where we can actually do future savings in terms of, automating stages of, say, phage susceptibility testing, as well as at the end for fill and finish of a phage product. And I think one of the comment was, advocacy, and I think it's very important because we actually do rely heavily on government funding to make this work. And from that perspective, we actually have done a comparative matrix of all the existing phage therapy centers that are actively treating patients at the moment. And if you actually have a glance over, almost all of them have heavily subsidy or government funding. And I think in the US, Singapore, and UK, you are slowly developing a kind of funding model. So you have a mixed government as well as the private partnership to keep this going and produce phage that make the patient accessible. From the regulatory side, because we are GMP exempt, and the STEM protocol, the beauty of it is that you do have the clinical oversight, and so you still produce phage to the best effort you can. And so, for example, with our products, we produce 4 reports. We rely heavily on genomic sequencing. We look at the manufacturing host, the bacterial host, to make sure, it doesn't contain AMR genes. The plasmid is okay. And then we looked at the genomic makeup of the phage itself. We have a batch report of the phage batch. And of course, we do susceptibility testing. And right at the end, we do sterility testing, as well as endotoxin testing, etc. So pretty much all the standard that you would actually place for a biologic.
Great, thank you so much. I'm cautious of time. I wanted to get a few more questions. We probably circle back, and it sounds like this notion of regulation and communication sandbox is an excellent way to move forward. It's also interesting in looking at some of the questions about the genetically modified angles that Australia has a unique perspective that may play into regulation in the future, which in Canada we don't necessarily have that sort of perspective at this time. But we won't go through the details of it. Danny, let's get to the next question, please. Phage biology would be a really leading question right now.
Okay, I can present my screen. I compiled quite a few phage biology questions.
Now, this is a phage therapy workshop / forum, so we'll see how many of these we'll take from a biology standpoint. Alex is also the president of Phage Canada and can tell me what to do. So Alex, let's see what you can do as well with these questions.
Maybe rather than I think I browsed a lot of these questions, and I think there's one really reoccurring one that I think could be a wonderful lead-in from Steffanie and then to you, which is how a patient actually accesses therapy, the logistics of it and how that has shifted. Because we're seeing a lot of questions from patients, I live in so and so, how do I talk to Greg? Or how do patients do this? And I think Stephanie's story, as a pioneer in trying to do this when there isn't a platform, but it isn't obvious to patients how they go about doing this. So maybe starting with Steffanie, how that process looked for her, and then where that is shifting to here in Canada.
Yeah, certainly. When I was looking into phage therapy for my husband at the time in 2016, there was nothing available. So we did everything from scratch. Now that we have IPATH, which was the first dedicated phage therapy center in North America, our process is that patients or their providers or their family members can email us. And the email address I'll put in the chat is ipath@ucsd.edu. We prefer to hear directly from physicians because we need to determine first what the bacterial pathogen is, and sometimes patients or their family members don't know. We also need to see an antibiogram. An antibiogram is basically the antibiotic susceptibility profile of that isolate. And if there are sensitivities to existing antibiotics for that pathogen, then the FDA, at least in the US, is not going to approve it unless there's a recurring infection that suggests that there's a biofilm, which antibiotics have a hard time penetrating. So that would be one of the few exceptions of that kind of a case. Now, each country and program is somewhat different. Canada, I'm sure Greg is going to talk about what the current process is, because in the West, phage therapy is an experimental treatment. So it isn't like you can just go to your doctor and get this. These are special cases, and people have to have run out of antibiotic options in order to get it. Now, after clinical trials are done that show hopefully that phage therapy is efficacious alongside antibiotics and is better or at least equal, then phage products can be licensed alongside antibiotics and be made more widely available. But until then, this process is going to be more cumbersome. And at this point, our center at UC San Diego really requires that there only be one bacterial pathogen involved in the case, because if there's multiple multi-drug resistant bacterial pathogens, phage needs to be matched to each one of those, and that gets very difficult. So it's usually a single infection, and sometimes we have phage in a library that's already characterized, already sequenced, ready to go, and it can be relatively quickly determined that, yes, this is a match, and then the phage needs to be manufactured, and the process for approval in the US, being the FDA, has to happen on an individual basis. But if we can't find phage in a library, then environmental samples like the waste runoff that was mentioned earlier need to be used, and that can take weeks to months. So it can be a very long process. Obviously, in Tom's case, we got very fortunate, but it can take some time, and for patients that are in acute septic shock, it may not be fast enough. So that's why we need more research. Greg, what's happening in Phage Canada? How do people get phage therapy here?
Yeah, so there's a special open label individual patient, which is a process that Health Canada has put forward. It is for those most serious conditions where there's no other option. Unfortunately, it takes as much work as a clinical trial to do one case. And having had to go through the process a few times, it is something where we really should be looking to develop a registry, which will have more of a streamlined approach. The STAMP protocol that we were talking about, or other protocols to make it so that people are in the study quickly and are assertively looked at through the study to allow them to get access to the right phage. Because there's matching that has to take place from your particular strain of interest, or there have to be a production of a more, let's just say, broad spectrum phage or phage cocktail that can work. though that takes time, energy, trust. And right now, and as Merrick was saying, like we're not even able to have the regular infectious disease service in the current framework because we're so busy addressing our situations with antimicrobial resistance and C. difficile that we need to look at smart ways of providing the resources to get to the next step. In the States in particular, they are overburdened, for lack of a better word, with these compassionate cases at the regulatory level where they're not able to focus on getting to the next stage, which is a randomized control trial. And in science in general, and medicine in general, is waiting for multi-center, multiple randomized control trials to really say phages work. We look back, over 90 years to show that in 1,450 cases, it worked 80% of the time. But that's just one off. We really want with a gold standard of proof. And I'm happy to say that there are at least two proposals in partnership with our colleagues in the United States and other countries, on one of which is on urinary tract infections, the other of which is on orthopedic infections. So we could be giving people a large recruitment opportunity to come into either orthopedic or urinary tract trials as early as spring of 2027. If we want to go faster, we have to look for private or philanthropic ways of moving forward and policy ways of sharing of what's going on. Horizon went ahead and put in 15 something million euros for up to 3 randomized controlled trials, and that will significantly help and improve things. Otherwise, as a clinical director for Phage Canada, and we're looking for an associate clinical director to help and navigate this process, I have a great interest in urinary tract infections. Obviously, we're expanding to orthopedic infections, and we have only 2 real main companies in Canada that are producing phages. One is in Gatineau, Quebec, which is called Keen Biotherapeutics, or biotechnologies rather, and the other is Cytophage in Winnipeg. And they have also been critical to the 10 cases that we've had to date and likely to be even more critical in the months to come. So it's going to take energy, time, and coordination. And it's one of the reasons why we have so many international partners today to try and help us move forward. I don't know if I answered the specific question. I'm happy for follow-up.
I think I can build this into a nice follow-up that maybe touch on you two first, but then broaden it to a wider audience. So there's a lot of questions in this about what the barriers are to phage therapy. And I think we've already touched on that quite a bit. I'm really curious as where barriers have really been falling down. What is some point in getting this forward where you've suddenly noticed, hey, there was a breakthrough here. When we got to this stage, it was a major change. And maybe starting with IPATH that has a little bit more history with this and where things stand. But then I think it's a great question to throw out to some of the other organizations that may not be as far or maybe much further, but they can point to a watershed moment where the landscape has changed. So maybe starting with Steffanie.
Yeah, I can mention what the barriers are, and I want to say first that these are surmountable barriers with funding and support and more infrastructure. We have a single coordinator, one staff person at IPATH, that, and so I mean that tells you we definitely need more support. But there isn't a single organized phage library, and so if somebody contacts us and says they have Burkholderia cepacia and it's multi-drug resistant, then I know who to reach out to. There's certain labs. I know which Dr. Van Tyne's lab has been very supportive in a number of different phage therapy requests that we get. And we reach out to labs and say, hey, we've got a patient with this bacterial pathogen. Would you help do a phage search? Sometimes the labs are overwhelmed and they can't do it. Then that becomes a problem. Sometimes we can't find a phage match or a suitable phage match, which are lytic phages that actually kill the organism. So in those cases, we either go to environmental sources or genetically modified or synthetic phages may be an option. And then there's manufacturing. Dr. Van Tyne's lab is one of the few labs that's actually able to manufacture phages. Sometimes we have a university lab that's, at Brigham Young University, Dr. Gross's lab has been very helpful, but they don't have the ability to manufacture phages because they're a university lab. They're doing this like pro bono. So we need to send the phages somewhere else to get manufactured. And I mean, grown up in sufficient quantity to treat the patient. And then we need these phages often to be clinical grade. So that means phage that's made with good manufacturing practices. And that's ideal because we want to treat phage like a living antibiotic. And it's not necessarily required by the FDA in all cases if it's someone who's going to die, but they want it as much GMP-like as possible. Now, these facilities are expensive. They're like giant bioreactors. That's where you're able to make sufficient phage in quantities to treat multiple patients. And so those are going to need like millions of dollars. And it isn't just one facility that we need. We need at least one on every continent to take phage therapy to scale, and ideally more depending on the needs of the population. So those are just some of the barriers that we've encountered. And but again, all of these are surmountable. I mean, this is a world where we had 4 or 5 COVID vaccines in 1 year, right? Where there's a will, there's a way. So I urge all of you to get active, talk about phages in your communities to your, the policymakers, and to vote with your feet. And we can make this possible. Greg, over to you.
Yeah, thank you so much. We're actually going to a meeting to talk to the Arthroplasty Society. Those are people who do revisions and joints, and to share with them more interests in trying to partner. So a lot of this is boots on the ground with groups that are already distinctly involved to try to make it so phages aren't feared and they're thought of a main issue. I also think there's a certain amount of urgency. And when I went to the UK recently, and they shared with me, because of the war in the Ukraine, that there are many, many people having injuries for a long period of time, that the resistance rates are mind-boggling, where usually for us it would be like 0.1 or 1 in 1,000 of a pretty dangerous organism. In some of their organisms, it's 50% of the time that it won't work for in Klebsiella situations for meropenem, our go-to antibiotic, or 25% of the time E. coli. So we need to be able to shout from the hills that we have to look for tools, and if it's endolysin, or if it's phages, or if it's other strategies, we're needing to look for that support. If we put together a 274-page proposal with 21 partners in 9 different countries on trying to just give phage to 220 women, and that is the proposals. We have the proposals, but we need the energy and the effort to make even stronger proposals with our partners. So that is ultimately, work over time is what we're looking to do, and the more that we can invest, the better. Yes, and Dr. Gina Sutt, thank you.
So to answer your question, Alex, I at least haven't come to that watershed moment yet where things seem to move ahead in a significant way. But I think that is coming. And I think that the 2 keys to this are massive cooperation, which is exactly what we're talking about now in this forum. But also, I think that, and by that I mean centralization. And many of you on this call have talked about that, centralizing, the phages. Because as Steffanie nicely said, when I get a patient, I spend 40 hours on it, and I'm a busy clinician, and I don't, I must be crazy to be doing this. And not a lot of people are out there doing it. So that's one barrier. But if there was a centralized way to one place where I can send my phage. And then I think AI is going to be the next big thing where we're taking AI and we're making matches, not empirically on the plate, which is what we're doing now, which has been a very solid and reliable way to do it, but it is inefficient, right? So I think, as Steffanie said, we can do this. There's a way to do this. But it will take massive cooperation and lots of money. So, yeah.
Yeah, maybe to quickly also add in, Jean-Paul Pernet, who's our famous Belgian colleague, wants to have a device in the future where we're actually making phages in a a method that doesn't require all the efforts and struggles where you put in the sample on one side of the swab and 8 hours later out comes the phage from a production standpoint. You can do all of this, but it takes a gigantic room to do it. But it's that interest of having a more capable thing. That machine obviously would require a tremendous amount of global development and also has to make it fundable. So we can't even fund regular antibiotics, let alone a machine to make antibiotics. So do we have to look at innovation in a different lens? So that's my long answer.
I would like to throw that out to some of the other country organizations. So we're maybe starting with France. I know we had the network from Lyon. If we still have, perhaps not, then we'll circle to Phage Australia before we go back to Tristan.
Tristan, you want to go first?
You can, please.
Oh, okay. I think my watershed moment, there are two of them. First is getting the human ethics approval through, and it's a national HREC for the stent protocol. But before that, we pretty much have kind of consultation clinical meeting to make sure that every site or every hospital, major teaching hospitals participate, and there is a principal investigator for those sites. And therefore, it's a top-down approach that, okay, we're doing phage therapy. And we came up with quite a few education package to teach the nurses. I don't know how many times I've actually baked carrot cake and muffins to go to those clinical meetings, try to talk to, convince them that phage therapy is safe, is tolerable, and talking to different clinicians. So I don't know if you're aware, we conducted quite a few surveys, and the first survey we did was Australian prescribers. And because we've got universal health, we wanted to know if in the public hospital, how the clinicians would view phage therapy as a clinical service. And through the results of that, and it went through 2 different iterations. Through the results of that, we were able to identify what the clinician deemed as the priority to treat. So in this case, it was cystic fibrosis, bone and joint infections, as well as patients that have gone through transplant. And from there, we're able to convince different professional associations to actually implement phage therapy. So that's one of them. The other one was because we've been engaging with our TGA, our regulatory bodies, and we have a very supportive state government. So in this case, New South Wales state government, and they were able to facilitate. So, I'm an academic, so I wouldn't have access to all these, top bureaucrats. And it was through them that facilitated and convened these regulatory talks that we were able to get through. We talked to the manufacturers, people that know about GMP, but they don't know anything about phage therapy. We were able to have a platform that we can discuss all of those. And so recently, last month, we actually have a modified Delphi process where we actually wanted to formalize. So there's a lot of paperwork beforehand. We met for one day and we went through 44 different statements of how we would regulate phage therapy. So this is the nitty-gritty of, if you have a MOI of such, would you actually then go on to the next step to select the specific phage for the patient? And for example, and the other example is, how many reads would you actually deem as when you go through genomic sequencing, that you would deem that suitable for a clinician to sign off and say, okay, this phage product is good. So those are the 2 watershed moments for me.
And then Tristan, to speak to when things change the most in favor of phage therapy, if there's a moment you can speak to.
In fact, in France, there was a a break in the approach of phage therapy based on the iterative discussion that we had with the French Healthcare Authority. But finally, they discussed with the phage producer in France. We were lucky because we had a company. And then they asked us to build for the patient care different things. First, they ask us if you would like to use phages, you have to use pharmaceutical grade phages, and they have put a list of things that are mandatory to has to be followed as quality control. And we follow that using phages from the industry first. And if they are not active, it is possible also to use magistral phages from the Queen Astrid Military Hospital. So finally, based on the discussion that we had and that we have with the French Healthcare Authority, we have both system finally. And it is also a discussion with each phage producer in each country, with the French, with the healthcare authority in each country, and with future users, and especially physicians.
All right, thank you. Now, Greg, if I'm not mistaken, we have a precious 4 minutes here.
Yes.
And we want to end on an optimistic note.
Yes, yes.
So perhaps, maybe throwing it first to you, and then you can throw it to other people. Like, what is the thing, that the one thing, not the long list, that people in this audience can go out and do to advance this? There's been a lot of chat in the Zoom chat about advocacy, about reaching out to MPs. Where do you say someone in this audience goes 5 minutes from now and writes a letter or does a thing or recruits someone? What is their step?
3 things. Have a conversation at the dinner table. That was simple. Talk about how we need to do this to fight superbugs for the future. Second thing, if you're a clinician, sign up for global clinical phage rounds, or if an infectious disease physician, sign up for the phage therapy working group and this global clinical phage rounds that Gina and I and others put together to talk about cases. And the last thing, everyone should sign up for Phage Canada, including the general public, where it is because of the 10% or so, 12 of 120 members that we're looking to further advance the field and have more sessions like this, and have it so it's maybe a smaller group, a little bit more of a chit chat, and also have it more organized so we can have it be an exception to this idea, well, we don't care about healthcare, or we're not able to coordinate something innovative. Ultimately, University of Toronto was successful in sharing a Canadian gift of $5 million, and there are other gifts that are taking place. So reaching out and having that sort of conversation further will be helpful. I also know there's probably about 30 questions that were asked that we were not getting to today, and I'm sorry, maybe we have to figure out a smarter way or better way of doing that. We hope to answer those questions in the future to give everyone the tools they need to notice that we're coming to a very important fork in the road, and I want us to rise to the occasion, not just for Canada, but for Canada's place to support our challenges about antimicrobial resistance in the world. So anyone else? I think what I want to be able to do, if that's okay, Alex, I want to make sure we do 2 things really quickly. We want to share the code for an evaluation where you can add more information. And if there was a question we didn't get to, let's go ahead and show it really quickly. I would also at the same time encourage everyone to come off of their video you know what I'm trying to say, come onto their video so we can have like a happy thumbs up, like let's move forward, but with determination, because people aren't happy when they're suffering from infections, and we need to work together to make that work better. Further to this, if you're on the evaluation form, what you'll do is you'll also get access to the video for today, and we can continue this dialogue through YouTube as it will be posted in the Phage Canada feed. So we have people that are now in this thumbs up position. Can we do the thumbs up? Okay, we're trying, folks. Thanks everyone internationally for all your support, nationally as well. Really appreciate it. Amazing work. More to come. This is just the beginning. And then the other thing, Danny, can you share the QR code for the survey and everything else? Okay, almost there. going to the end. Dr. Steffanie Strathdee, your chance for final words. And, I'm also happy to share that you got the Impact Award from the Faculty of Medicine, University of Toronto. Amazing work, and it's more impact like you that will help us get over those barriers and get it through that fork in the road.
Thanks so much, Greg, and to Alex for inviting me to speak. I think I want to end by saying that, the counterfactual of not, having phage therapy available is that one person every 20 seconds is going to be dying from superbug infection between now and 2050. We can't let that happen. We know that phage is just one tool in our arsenal to combat antimicrobial resistance, but it's nature's own, weapon, and we can harness it to our own good. So let's do this, and let's keep the momentum going.
Okay, thank you very much, and let's keep the momentum going. You are seeing a poll. Here's your last second. We're only going to take 30 more seconds of your time so we can get this information. Alex, in those 30 seconds, anything else you'd like to share?
I want to in particular reach out to members of the general public and patient populations. Phage Canada and many of these organizations tend to be somewhat insular within the community, of practice, of care, within the community of research. And the more that we can get out of that box and reach out to the general public, and the more that the general public feels that they can come to these communities of clinicians, of regulators, and talk directly to those people, I think that's going to be a major step forward. So hopefully we can continue to do better on that front.
Okay, we try to stay on time in our world. You can see the results. We have 68%, which I believe is a good increase. 20 for very familiar, somewhat familiar, also increased. So I think we did our job today. We look forward to the evaluations, and thanks for signing up for Phage Canada. I'm going to sign off now. Take care, everyone. Thank you, Danny, for everything and try to make this happen, and amazing global teamwork. We look forward to Phage forums in other countries in the future, and Canada will be standing by to share what we've been doing. Bye for now.
Thanks, Greg.